I'm very pleased to introduce Dr. Chris Halbrook, who's an assistant professor in the Department of Molecular Biology and Biochemistry in the UCI School of Biological Sciences. And he's going to talk a little bit about the work supported by the Anti-Cancer Challenge on pancreatic cancer. Dr. Halbrook.
Hello, I'm really excited for this opportunity from the Anti-Cancer Challenge to be able to share some of our research in my lab with you, and Dr. Van Etten for allowing me the opportunity to do so. I just wanted to give you guys a quick background of why I'm here. I'm a new assistant professor at UCI. So I've worked in pancreatic cancer ever since I started my graduate training at the Stony Brook University in the Mayo Clinic. And this was about 10 years ago. You can see me in the background here back when I was still full of youthful vibrance and a little bit more hair. And then I transitioned to the University of Michigan to join what has really become one of the largest pancreatic cancer research teams in the country, where we were really pushing the envelope on a daily basis in terms of how we're discovering cures eventually for this disease. But when I was looking for the place where I would go start my own independent lab, I realized that UCI was a very set up to be a very good place to do groundbreaking research, but we didn't have anyone that was focusing on this disease at the time. So this, I think, is going to be the next, hopefully, big site that we'll see that the West Coast will have its own contribution to where some of the other larger groups like the University of Michigan, MD Anderson, and the groups in Boston have really been the center of leading the field in terms of this particular cancer. So why am I focused on pancreatic cancer? So this is a list of different cancers that people are expected to be diagnosed with, projected up until 2040. So pancreatic cancer is actually way down here. So you may think to yourself, this isn't that big of a priority. But if you look at the number of people who are actually dying from cancer, you can see that pancreatic cancer is sitting here right now. So we're about here. We're currently the third leading cause of cancer-related death. And this is projected to become the second leading cause of cancer-related death by about 2030. So we're getting less people are dying of other cancers you're more likely to be diagnosed with because we're getting so much better at treating it. But when it comes to pancreatic cancer, we're getting around the same number of people with a progressive increase, but not exponentially increased. But everyone who gets the disease so far is not faring very well. So again, I've been in this particular field now for about 12 years, and this is the first presentation I ever gave. I went and pulled up the introductory slide for this. And at that time, the 5-year survival rate for pancreatic cancer was 5.5%. We didn't really know the major causes of the disease other than smoking and chronic pancreatitis. And at the other end of it, there's which is now, I think we realize about 10 to 20% can have familial pancreatic cancer disease causes or underlying genetic predispositions to it. But that means about 80% of people who get pancreatic cancer, we don't have any idea why they're getting it. And when we're talking about pancreatic cancer, I'm talking about a disease called pancreatic ductal adenocarcinoma. So this is where we moved to in the 12 years I've been in the field. There was a big goal where there wasn't originally a lot of funding for this disease because it was considered essentially a death sentence. So why would Congress want to put money into this? So in the early 2010s, there was a passage of an act called the Veltricicant Cancer Act, basically saying that if we have diseases that are this deadly, why are we not putting more money into them? And so there was a goal over the 2020s that we were going to double pancreatic cancer survival from this 5% figure by the 2020. And so we've been able to achieve that. And we're still progressing up slightly per year, but obviously we're still failing the vast majority of patients. But this level of the disease that was originally 5.5% had stayed at that for the previous 30 years. So we're really finally starting to make some progress in treating this disease. So I had a really unique opportunity recently where I was approached by one of the flagship journals that publishes biomedical research called Cell. And they asked me and several colleagues of mine from the University of Michigan and one of the pioneers of the field from the MD Anderson Cancer Center, what we really think have been the biggest things that have changed in our understanding of pancreatic cancer. And so in this time frame, we've really seen an idea of knowing exactly what the mutations are that drive this disease, how the cells in the pancreas, which cells are the ones that are actually mutated and will lead to the disease, the precursor lesions that we could hope to eventually screen for, and then billions of dollars that was spent sequencing tumors in order to figure out that there was various subtypes of tumors, which unfortunately so far have not actually really informed how we can treat different patients. The other thing that has become abundantly clear as we continue to research this disease, so this is what we call a hematoxylin and eosin section. So this is a slice of a tumor that we stain with a couple different dyes to try and just see the architecture of a tumor. And what I'm bringing this to your attention for is this thing right here, this little island of these particular looking cells. These are the cancer cells in a pancreatic tumor. The rest of the stuff that's not these little islands is actually stuff that is not cancer cells. So there's other cell types in there called fibroblasts, there's immune cells, and there's a lot of this is just filled up with extracellular matrix. So things like collagen that make the tissue really stiff. And it's one of the things that a pancreatic surgeon can tell you when they take one of these tumors out, is that this fibrosis of the extracellular matrix is essentially scar tissue. And these tumors are eventually as hard as a baseball when they get taken out. There's so much of the scar tissue that's in the tumors. So what we've actually, and others, is really a big focus of the field, have done is looked at if the cancer cells aren't the major cell that you actually find in the tumor, what are the other cells in the tumor doing to actually shape how these cancer cells are getting their signals to grow? And so there's been, again, just an absolute mind-blowing amount of heterogeneity that we found. So different cell types within individual cells that we found. So within the things that I was introducing that are fibroblasts, we found that fibroblasts are not just fibroblasts. There's actually at least 3+ different types of fibroblasts you can find in each region of a tumor. These are different between patients. There's different types of immune cells that exist within tumors. And each one of these lines indicate different interactions that different studies have shown that can be targeted therapeutically to potentially take apart this crosstalk network between the different cell types that support the cancer growth in the tumor. And the other thing that's become abundantly clear with these lines this way is showing you that these are interactions that are actually inhibiting the immune cells that you'd like to see in a tumor. So these CD8 T cells, these are the cells that would respond to immune therapy. You do not find these in pancreatic tumors, and these are one of the tumor types that just do not respond to immune therapy as much as you've seen this be a miracle drug in some other tumors like melanoma on occasion. So again, the amount of different heterogeneity between the cells, so the different cells within an individual type has been just one of the biggest things that the modern technology that we're allowed to use in research versus the technology we had 10 years ago is really uncovering. We can see different cell types that are in a normal pancreas that expand into different cell populations in the tumor. And we found that some of these are actually beneficial for the tumor cells, so we want to get rid of those. But some of them actually have roles in preventing the cancer from metastasizing, so we don't want to get rid of those cells. And there was classic clinical trials for pancreatic cancer in the early 2010s where we didn't know there was this many different types of these fibroblasts that exist within a tumor. And so targeting all of them actually let the cancer metastasize faster, which in a tumor that kills as fast as this does, it was really a sad result for a clinical trial. So we've really come up with an idea of what we consider the way that we're going to eventually cure this disease. So we know the mutations that drive the disease because we've sequenced it. There's finally over 30 years, people have been trying to target this particular protein. And in the past 5 years, we've seen compounds finally be developed. In the past 3 years, they've been deployed in the clinic. And now we're finally seeing the data that is coming from patients being treated. It looks fantastic. We know that there are patients that respond to other therapies because they have these familial drivers, which again, years ago, we were unaware of. There's specific features of pancreatic cancer cells that we can target. All the other cells in that tumor microenvironment, we call the surrounding area around the cancer cells that support the tumor growth, how we can leverage the interactions that are in there to block the ability of them to suppress the immune response that we want so we can actually use immune therapy in these tumors. And eventually we're going to translate these into innovative clinical trials so we can get patients into trials that work for them faster as opposed to just having them sit on trials that aren't working for them. So I focus more on basic research in which my lab focuses on the mechanisms by which these parts of how we're going to eventually cure pancreatic cancer are focused. and how these different things will work together. So over the past 7 or so years, we've actually focused a lot on not just how cancer cells can communicate in terms of signals they exchange, but also in terms of the way that they share nutrients. And we found that this has incredibly profound effects on different aspects of the tumor biology. So those fibroblasts I was talking about, these are cancer-associated fibroblasts, can feed pancreatic cancer cells certain nutrients. We see macrophages, which can be about 40% of all the cells in a tumor, can directly block the therapy that we use for chemotherapy. We've shown mechanisms by which these cells get programmed. We can see the cancer cells can actually eat that matrix that I showed you. We can see there's different types of cancer cells that act to support each other. And eventually, we've also shown now that these macrophages are directly blocking the ability of these T cells you want to come into the tumor by degrading a nutrient that they need. However, almost all of the work that we've done so far is focused on what would happen in a pancreas tumor. But when most people are diagnosed with this disease here, they don't have this localized disease. Only 12% do. If you have localized disease, your 5-year survival rate is much better than the people who get diagnosed with metastatic disease here, which is about over half of the patients. These are the patients who do the absolute worst. And so these are the patients that we actually need to focus on treating. So what we've done through the money that we've obtained through the Anti-Cancer Challenge is we've used mouse models that we've developed for pancreatic cancer to ask the question is what is different with a tumor that is in the pancreas to what happens when it metastasizes. And the liver is the most common place you see a tumor metastasis in pancreatic cancer. And so this is working with a good collaborator at the Henry Ford Pancreatic Cancer Center. This work's done by a very talented graduate student in my lab. So we're using a technology called single cell sequencing, which allows us to look at all the genes and how they change on an individual per cell basis. This is one of those things that has really just revolutionized how we think about different cells in a tumor in the past 5 or so years. So these data are very complicated. So each one of these dots is a cell that we have the entire expression level of every single gene on. So this becomes a big problem of trying to figure out computationally what to do with this much data. But we've developed ways to be able to identify which each of these individual cell types are. This is a visualization that as they sit closer together, they have shared features, which we can eventually then say that, for example, these red cells that you find in both the pancreas and liver tumors are those fibroblasts that I was talking about. We can see the different proportions of the cells that exist within these tumors. We can actually look at within the cancer cell populations themselves. We can see each one of these vertical lines is a cell, each one of these horizontal lines is a gene. When you see these blocks of yellow, these are shared features that we're allowed to say that these are able to say that these are different populations of cancer cells, which have different features that we can target with different drug types. And one thing that was very important for us to find with this is you can see this green bar in the liver tumors accounts for over half of the cancer cells versus the pancreas tumors. And the ones that are these green ones here have very specific pathways that we think we can target with different compounds that we've accumulated to do this with. And the last population I wanted to highlight that was very interesting for us. So there's this big focus on these cancer-associated fibroblasts that are in the primary tumors. But what we found so far when we're looking at our liver tumors in comparison is that there's these pink cells right here that almost entirely are located in the liver tumors here. And if you look at this distribution, almost half of them in the liver tumors don't fit any of the things that have been described with every thing that people have looked at with the primary pancreatic tumors. So essentially the biology of the liver tumor is completely different than that whole network of interactions that I've been showing you before and is still relatively undiscovered. So we've shown these fibroblasts are real. I won't have time to get into it. We're working with a really talented investigator in the developmental cell biology department here who's developed these algorithms to predict the interactions between cells, and we can see our cells of interest, how they are talking to other cells, how cells are talking back to them, different networks that we can use to target this. And again, this is done in collaboration with the postdoc in Max's lab, Ying Zi. So again, this is still early days with this, but that's the whole purpose of this mechanism is we've generated enough data now that we're putting in grants for long-term studies involving this whole thing. But we've also seen so much that there is left to discover in this, and this is only possible through funding through the Anti-Cancer Challenge. So with that, I just want to quickly wrap up to thank the people who are in my lab right now. Again, this work was largely done by PhD student Rima Singh. We've been very fortunate to have our work funded through several mechanisms. Again, most important for this project is the Anti-Cancer Challenge. And then we also have our lab and myself Twitter accounts that you can look to actually see different things that are coming out of the lab on a regular basis. So I encourage you to also follow us there. But with that, I'm happy to also answer any questions that you have on this and other things to do with pancreatic cancer as well when we have our Q&A session.